Wonder, where are they? Is it group one? So where are the group one people? Where are they? They should sit. They should sit here. Anytime your group is presenting. Okay, you want them to stand with you. Where are they? Group one, where are they? Eh? Where? Only you I'm seeing now. Let them come. Where are group one people? And they should introduce themselves. Group one, introduce people in your group. Who is the leader of the group? Who is the group leader? Eh? Eh? So you are standing in for her. So introduce people in the group first, and then we now start.
Don't mention their name. Don't mention their name.
Tell them.
Just speak. Like okay, good afternoon, everyone. So on behalf of Group 1 members, we have Miss Juma and Mr. Great to present for us. And we are working on investigation of the impact of climate variability on malaria incidents using machine learning techniques in Nigeria. Thank you.
Good evening, everyone. My name is Junaid Muslima, one of the representative of Group 1. I'm here to present our research work titled Investigation of the Impact of Climate Variability on malaria cases using machine learning techniques in Nigeria. Malaria remains Malaria remains one of the major public health challenges in Nigeria because it carries the highest malaria global incidence. Malaria transmission is climate sensitive because there are some factors that malaria transmission is climate sensitive. There are some factors that influence it, such as temperature, wind speed, solar radiation, among others. So, okay. And these factors influences mosquito breeding, their survival, and parasite development. The relationship between malaria and climate is complex. Therefore, machine learning provides an opportunity to identify these complex relationships and provide historical climate and malaria data. Malaria control remains largely reactive in Nigeria it is reactive in the sense that malaria action control are not taken until there's an increase in malaria incidences. So the aim, okay. The aim, so the aim is to investigate the impact of climate variability on malaria incidents and to forecast incidences using machine learning across Nigeria's geopolitical zones. So specifically, the objective, the aim is to determine the relationship between climate variables and malaria cases and assess the multicollinearity among the climate predictors across the 6 selected states. Also, it is to develop and evaluate machine learning models that will be used to forecast new malaria incidences, that is 1, 2, and 3 months ahead using lagged climate and malaria case data. Also, it also compare predictive performance and generalization using the linear regression, SVM, random forest, XGBoost, and other MLP models across the 6 geopolitical zones. The research question include which climatic variables are significantly associated with malaria incidences in the 6 selected states. Also, which can machine learning models be trained on lagged climate and case variables to forecast monthly malaria incidents 1, 2, and 3 months ahead with acceptable accuracy? Which model, linear regression, SVM, random forest, XGBoost, or MLP neutral networks generalizes climate transmissions across the 6 geopolitical zones? Previous studies have been carried out concerning these topics. However, most of these topics are geographically limited. It is on this basis that the study aims to investigate the impact of climate and malaria incidences across using 6 selected states in Nigeria to represent Nigeria geopolitical zones. Mr. Grace will continue from there.
Yeah. Okay. Yeah. Yeah. Good day, everyone. The volume. Okay. Good day. Are you all hearing me? Hello. You can hear me. Okay. Okay. Good day, everyone. In continue. Okay. No. Sir, just leave it this place. These guys. Okay. For me to be thinking, I'm putting it slow. Hello. Okay. Thank you. So I'll be moving through the problem statement. First of all, we are working on the investigation of climate variability among Nigerian cases using machine learning models. So in the problems, we can clearly see that malaria control in Nigeria have remained largely reactive with intervention up to deployed after increases. What that place is telling us that malaria cases, we in Nigeria, we often deploy intervention when the thing has happened. We are trying to cover the gap. According to Nuclear Parasite and Climate, they said Nigeria lack an operational climate-informed earlier warning system because we don't have a system that can help us to know what is going to happen in the next 2 days. Just like Professor Chong was talking about yesterday, a system that can predict what is going to happen. We're supposed to have a system. So in this case, we are trying to look at how the study is going to examine the gap by using we are using each state. The reason why we pick each state because we want to know their climate conditions. We decided to pick 6 states from 6 geopolitical zones to understand how climate conditions work in Nigeria, whereby applying machine learning models. And we are doing a supervised machine learning technique because we are working on trying to find the patterns and also trying to understand non-linear relationship. And also we are trying to also forecast that in the next 3 months, for instance, Ekiti states I'm not saying it's true, but for the next 3 months, there might be like 20,000 cases of malaria, whereby the doctors, the hospital, they get informed early 2026 now, that the next 2 years, which is 2028, that there's going to be 20,000 cases of malaria. So I'll move straight to the related work. All these people, I'll just summarize what they did. What they did is just like they focus on generally 6 countries, focusing on informatics in medicine on log, in predicting malaria outbreak for meteorological data using random forest. None of them actually focus on 6 geopolitical zones in Nigeria. That was what make our study different from their work, whereby even Zante 2015 said systematic review of malaria forecasting model, most study use regression or anemia. So we were trying to have a difference in their work in the sense that we are going to use some kind of mathematical model, some kind of machine learning model with statistical model, in the sense that when we bring out the results, we are also going to project the results for future forecasting for the 3 months. So I move next to the gaps. I said it over before. The established association, not operational forecasting. That is what they did. They did not do operational forecasting. None of them never forecasted for 3 months. They were just using Nigeria as a whole. That okay, this is what we have. This was based on the articles that we got. Now I'll be moving straight to the methodology, which is our study location. For you to have a good research, you must always have a good location, which is also known as population of the study. What your study is going to focus on. Now our study is focusing on the 6 geopolitical zones. We have Kebbi and Kano, Benue, Anambra, Bayelsa, and Lagos. This map was generated using GeoPandas Python library in Python. We use it to generate the map in the sense that we're going to have some color graded representative. That this state, I don't know if there's a way you can zoom it, sir. Can you zoom the state, sir? In the sense that those places that you see that are highlighted shows that these are the areas of where our study is focusing on. Thank you very much. You can see Kebbi Northwest, Lagos Southwest, Anambra Southeast, Bayelsa South, Benue North Central, and Kano Northwest. So I move over to the next slide. Okay. Sorry. So our source of data. We got our climate variables. We use temperature, rainfall, wind speed, and solar radiation, which is also known as all-surface radiation. Because when we went to some websites don't really have solar radiation as a whole, they call it all-surface downrange radiation. So we got it from NASA power site for each of the states from 2015 to 2021. The same thing with the malaria incidence data. We got it from NEP, National Association of Malaria Repository from NEP, which is Nigeria Elimination Monthly Program. We got the data from there for each of the states from 2015 to 2021. Now, you can see some of our states, the Anambra, the Bayelsa, Benue, Kano, Kebbi, and Lagos, geopolitical zone, Southeast, South. How did we get our monthly mean cases? We added all the mean. Can I get a marker to show some calculation? Okay, time has this 5. Okay, let me just say that we got our mean monthly cases where we added all the means of January 15 to January 21. These 5 minutes here, so I see after. No. Okay, then our data processing, we're going to be doing chronological splitting in the sense that we're going to train our data from 2020 to 2021 and test our data from 2020 to 25. Okay. Then we have our modern technique, linear regression baseline, which is going to be our baseline for us to use other models, random forest, which is going to allow us to get many decision trees, support vector machine, which is going to allow us to predict observation and use RBF kernel to analyze a non-linear regression relationship. Then we have XBoost, which is also a tree-based model. We have MLP, a neural network. Sir. So our future direction is that based on what we have done, we are going to have a more data processing, a model fitting, a comparative analysis, a forecasting, and a manuscript preparation that will enable us to bring out data and be able to help health sectors to predict malaria cases in Nigeria.
Thank you. Yeah.
Okay, thank you. Group 1, you have tried. Let's clap for them. Now, this work you have started, I saw that. Can you go to your research question?
Sorry, can you hear me? I know. And we keep. It's not even work. Wait, it's not moving. Sorry.
Okay. If you look at your research question, you'll find out that you have mixed them with methods. Can you see? So you need to look at that and come out with questions that are relevant to the epidemiology and climate variability of malaria. How climate, and then I want to also believe that your title is not the final one. You are still working on it, right? So because the word investigation looks very sterile, but that's supposed to be an objective. Do you understand me now? So you have to look at that. So, and then I don't know, what deadline should we give you to finish this work? Yes, we are very serious with what we are doing. Deadline, because I'm in that group, that your group one. So I have to start. So I told you that I'm going to be a monitoring spirit. So what deadline should we give you? Yes, today we are in August. Today is August what? August 14. So when should we expect the full paper to complete? Group one. Yes.
Okay, so this is what I will see.
Okay.
I will just see if you can give us the deadline to next week Wednesday.
No,
Now, if
you are joking. His publication, do you think is okay? You want ChatGPT to write for you, have you?
No, sir, that's not okay.
ChatGPT produce
No.
paper.
No, that's not.
Okay, so tell us. No. I want it here now. No story. Yes. We are in August, September, October, November. By end of November, I want to have this paper. November 30th, latest. I'm writing it down. Thank you. Next.
Thank you very much. Can we celebrate group one for their presentation? All right. Group two members can come here, and I believe they have sent their slide. So who are those representing group two? Okay.
Presenter, please. Yes, to the question, he was talking about machine learning. And when it comes to machine learning, we have a lot of field in machine learning. And we're talking about climate to malaria. In that machine learning, what method do you want to use actually? Because you are going to get a lot of results. So specifically in machine learning is a wider field. But what method do you want to use for the climate and the malaria? Give us an elaborate on that. I mean, you understood what I'm saying.
All right, thank you. I mean, you want to say method, machine learning model, I think. You know, in machine learning, since we have baseline, we need to check the pattern so that we know maybe it is a linear or what's it called? Non-linear. So all these methods that we are using, SVM, SGBoost, they are non-linear because we've viewed the pattern and it is non-linear. So we are using non-linear models. So we are checking each of those models, the one that will give us the best result. That's what we are going to present. Thank you.
Group two. All right. Group two. Group three. All right. Group three. Please, your slide is not group three. Group three. Okay. Group two, group one, you can climb the podium while they take your picture together with your. So group three, group two, please. It's ready. Group five. It's ready. All right, group five. So let's take group five. I think group five is ready. So, but don't like scattering disorder. Yes. Yes. Group 5. So group 5 members can come here. Then while we have your presenters. Oh yeah, group 5. Oh. Let's make it snappy. Yeah. All right, so we are the presenter. Where are the presenters?
No, sir. Okay, so I'll be inviting Mr. Matthew and Miss Joy to present. So this is this group. So Miss Joy. Joy to silent. All right, Miss Matthew and Miss Joy should come forward. Thank you.
Good afternoon, everybody. So I'm one of the representatives for group 5, and our topic is about antibiotic resistance in the community. So this is our introduction. Antimicrobial treatment contributing to the prolonged illness, increased healthcare costs, and morality. The burden is particularly high in Africa, where resistance among major gram-negative pathogens, including Ehrlichia coli and Kesai pneumonia, exceeds 70% of some antibiotic classes. In Nigeria, AMR was estimated to be associated In Nigeria, AMR was estimated to be associated with approximately 263,400 use laptop. There is no single intervention that can eliminate antibiotic resistance. Control requires coordinated measures across human, animal, and environmental health. Prevention includes infection prevention and control, safe water, sanitation, hygiene, appropriate vaccination. Diagnosis and surveillance support identification of resistant infection and appropriate treatment and antimicrobial stewardship promotes the use of antibiotics. So these are some of the literatures we reviewed. Go down. These are some of the literatures we reviewed. And we'll be going to the research gap now. Okay. the research gap. Consequently, there remains a critical gap in understanding how antimicrobial evolution and transmission between settings interact to drive the emergence and spread of antibiotic resistance. This highlights the need for an integrated modeling framework that combines evolutionary and epidemiological approaches. So the research aim, these studies aim to investigate the evolution and spread of antibiotic resistance. I'll quickly go to the questions. This study will address the following research questions. How do antibiotic misuse and overuse influence the evolution of antibiotic-resistant bacteria in the community? And how does environmental contamination with antibiotic-resistant bacteria contribute to the spread and antibiotic resistance in the community? So I will hand over to my colleague now.
So, all right, so based on the research questions that we have above, which are to investigate the effects of antibiotic use, misuse, and overuse on the evolution and the likes, we developed a compartmental mathematical model to investigate the effect of antibiotic misuse and overuse on the evolution and spreading of antibiotic-resistant bacteria in the community. And secondly, also we also did a data decomposition to uncover hidden temporal patterns in antibiotic use and resistant data by spreading the observed patterns into trends, seasonalities, and residual components. Now, let me quickly go to the compartmental diagrams. So our population we focused on the human population, and okay, thank you. So this is the schematic diagram. Meanwhile, HST represents the human population that has the that have the sensitive strain of the bacteria. Then at a rate of psi AB, they migrate to HE, which represents human population that has been exposed to the use of antibiotics in order to mitigate the effect of the bacteria. Then progressing to HT. Now they are progressing at the rate of rho. Now progressing to HT now, we are assuming that the antibiotic administration has cleared off the bacteria from the body of the host. Then as a result of that, there are also some populations that the antibiotic didn't clear it off completely. So they evolve into the HRS, which represent the human population that formerly has the sensitive strain, but has evolved into the resistant strain. And so HRS is representing the human population with the resistant strain of the bacteria. Then we have the SR, which is representing the susceptible population to the sensitive strain of the bacteria. Then we have the ER, which is the environmental reservoir. And the environmental reservoir, or which we can call the concentration of the antibiotic resistant strain into the environment due to contact and fecal contact, ready to eat food. So now the susceptible population interacting with the environment can also contribute to the human population with the resistant strain. So we developed this compartmental diagram in order to answer some of these questions. Can we progress to the next slide, please? Okay, so the next one, please. So we have a system of equations, 6 systems of equations, so expected outcomes. At the end of this research, we expect to be able to quantify the effect of antibiotic misuse and overuse on the evolution of antibiotic-resistant bacteria, including the condition under which antibiotic resistance is expected to increase or decrease in the community. Just like you can see from the compartmental diagram, that what majorly causes the bacteria in the body of the host to develop into the resistant strain is misuse or non-compliance of the host to administration of antibiotics. So when the host didn't take their drugs as they ought to take it, not all the bacteria will be cleared off the body of the host. So some of them will now evolve into a resistant strain. And that resistant strain continue to grow, reproduce, and multiply, and which now due to further treatments of antibiotics can make them either get treated or can it make them either get treated or still remain as part of that compartment. So this proposal has a timeline which we have put forth together, and we said by March 2027 tentatively, we should be able to complete the project, and by September, October, we should be able to enter every other thing. So here are the references that, here are the references that we have for this research work, and we also look forward to further improvements on the work. Thank you.
All right, so let's appreciate. Okay, wait. I'm going to take one question, then Prof. One question, just one. Okay, one. Okay, brief.
Thank you. I want to thank you for the presentation so far. You have done well, but like Prof said, your packaging. When you look at your slide, you have done a very good work. But when you look at the introduction and the next ones, they are not being compatible. You understand? Please try to find someone for your presentation. Are you getting it? Then the last but not the least, you have talked about your timeline. Your research gap is okay, but your timeline, May 2017, is too far. Please work on that. Thank you so much.
This man is talking like an authority. Are you an authority, AMR? No, Allah. Now, you want to respond, I mean?
The timeline is not actually far, but we are looking at how to get data. So if we can get availability data, we are certainly sure by on or before November would have been done.
Thank you very much. Now, somebody think that it's for now. You didn't talk about your data, but you jumped to data decomposition. So when you don't have a data, yes, you should. You don't have a data. You don't even know the kind of data you will get. So now this is an issue, and you didn't even also talk about the location you have in mind. Now, do you have an understanding of the geographical distribution of or geospatial distribution of AMR in Nigeria? Do you have the light? Anybody? Have you done any study on AMR geospatial distribution? So I think that's the first place to start. Now you need to understand what are the various locations that people may have done. You go to PubMed, you write this down, go to PubMed, find out where are the locations in Nigeria, geographical distribution of AMR in Nigeria. So if you know them, from there, you'll be able to know where are the high bodies of AMR. From there, you'll be able to know high body, medium body, low body. You'll be able to get that. And then if you get that, now you now begin to look for data. You can get data from PubMed, already published paper, right? And then you can also get data. I don't know whether NCDC, is there anybody working with NCDC here? NCDC. Did they, I'm not sure whether they I don't know to answer that question.
Actually, I don't work with NCDC, but I have a close relationship with them. And because of this work, we also put a call across to them, and they assure us that they have data to carry out this work.
And then we can look at their dashboard. Do they capture that kind of disease? Look at their dashboard and look at, on that dashboard, look at disease A to Z. They will write A to Z, disease. So if you see disease A to Z, look at it. Is AMR part of disease? If you see there, it may be that they are capturing it.
National health regulations. We have so many areas, and I'm part of the international health regulation, but my own focus is just on biosafety, biosecurity. So in our usual meetings, AMR is captured under IPC, that's Infection Prevention Control, and AMR2 has been prioritized as one of the diseases in the country.
Yes, there is a lot of discussion around it in the recent time. So try to start from geographical distribution of AMR. and then look at PubMed. What are the work that has been done so far on AMR? And then you can begin to look at the data available, and then you can contact some of these. Then I think if I will check, I don't know, if I check, I think I have one time downloaded the National Strategic Plan for AMR. So I think I have it. If I see it, I will send it to the platform. So, but you need to go look at, those are the things you have to look at. From that strategic plan, you can also get information on where you can get the data from. So I don't know the kind of data you get. The kind of data you get will determine whether you will be able to do some of the things you put there. You understand? Whether it is going to even be continuous time model, or it's going to be something that will depend on age or other things. So, but we appreciate what you have done so far. You can still, there's still room for better improvement. But on the issue of deadline, we have to talk about it. Now, you said March 2027, and we are in August. It's too long. Okay, so maybe let's give everybody general deadline instead of saying this thing. Yes, I give you general deadline, and it should be by December. So I'll change it now to December 15, so that before Christmas you submit your paper. December 15, everybody, please. So let's target that. So let's target that. Thank you very much.
Thank you very much, Prof. All right. What's group next? Which group is ready next? Group 2. Okay, group 2. Group 3. Oh, photograph. Thank you. First for group 5 members. Let's okay, group 3. Group 2. Group 3. Group 4. Group 4 is ready. All right, so let's welcome group 4 in the front and their representative. Group 4. Let's have group 4. All die and I die. Representative, you are okay. Just only you. Just only you. Is it just only you? No. Don't. I called group 4 already. Don't worry. You have to. I called group. Okay. Okay. All right. Group 3. Let's. Huh? Group 3. Okay. Group 3. Let's have group 3, please. I'm sorry for the confusion. Group 3. Then we'll welcome group 4. Group 2. Please ensure you send your group 3. Hey. No, I want to know what I said. 50% photo charge you are. So that's the question. So group 3, who are the people representing group 3 for their presentation? Please come forward. So okay, let's take group 4. Let's take group 4. Group 4, Miss Happiness, 10 minutes. Group 4. Represent, sorry, for. Yeah. Mr. Aaron, you want to introduce your, okay? Your representative.
All right, so thank you very much. So this is the group 4 for the fourth Ikamda school, and we've worked on thus far modeling the interplay between antibiotic usage, bacterial fitness cost, on the emergence of resistance in Nigeria. So we have two of our group members who will be representing us in this presentation. Thank you very much.
Good afternoon, everyone. I'm the representative for the Group 4 group. We are here to talk about the model. We are here to model the interplay between antibiotic usage, bacterial fitness cost, and emergence of resistance strains among bacteria. So we are not foreign to what antimicrobial resistance is. A group just presented on that. So next slide. So we know antimicrobial resistance is widespread. There has been enough information about this going around. A lot of work has been done on how we can curb the menace of antimicrobial resistance. And we know that most of the implicated strains are the gram-negative bacteria. And ideally, in antimicrobial resistance, we usually have multi-drug resistance more often than not. The resistance is not always spread to one drug. It's usually to multiple classes of the antibiotics that is being popularly used based on the WHO classification. And we also know that the factors that affect the emergence of these strains are the cost of biological resistance, which is defined as is a measure of the fitness of the bacteria. That's the ability of the bacteria to reproduce, and the virulence and the ability of the bacteria to reproduce under ideal conditions. We also know that compensatory mutations are a huge factor in how the bacteria handles the fitness cost. And we also know frequency and use of antibiotics, which is one of the primary factors that induce antimicrobial resistance. And we know that resistant mutations has a fitness cost on the bacteria. The bacteria is unable to carry out essential life functions by the presence of the genetic material that helps the mutation. So next slide, please. Next slide. So we know resistant mutations have minimum impact, and they offer a fitness benefit depending on the genetic and environmental conditions. So we are here to really talk about how does the fitness of the bacteria. A lot of work has been done on the impact of antibiotics on the emergence of resistant strains. And we are here to talk about how does the fitness of the bacteria, how does it also contribute to the emergence and persistence of resistant strains. So the aim of our study is to look at the interplay between the theory. How does antibiotic usage and fitness, how does it affect the spread of antimicrobial resistance? How can we tap into that to suppress the emergence of resistant strains? And our research questions are, we are asking the questions of effect of antibiotic usage on the emergence, persistence, and suppression of antibiotics resistance in Nigeria. And we're also looking at how does bacterial fitness contribute. A lot of work has been done on antibiotic usage, and we're trying to introduce a new parameter. We're looking at how bacterial fitness contributes to that. We're also looking at the combined effect of both antibiotic usage and bacterial fitness. So I will be demonstrating an analogy because I don't think a lot of work has been done around fitness. So when we talk about bacterial fitness, we are talking about the fact that the ability of a bacteria to reproduce and to exhibit its virulent characteristics, it's a subject of how much the bacteria can actually do that. We are saying that in ideal conditions, the bacteria can have an ideal fitness, but in the presence of some external factors, such as antibiotic use, and then it affects how the bacteria can develop resistance to some certain drugs. So we will proceed to the research objectives, which is outlined to determine effect of antibiotic usage in the emergence, persistence, and suppression of antibiotic resistance. We're also looking at the influence of bacterial fitness on the emergence, persistence and suppression of antibiotic resistance. And we are looking at the combined effect of both the usage and the bacterial fitness on the emergence of resistant strains. So at this point, I would hand over to my colleague to continue with the rest of the presentation. Thank you.
Okay. Wait. Okay, thank you so much. So a lot of biology has been given already about the antimicrobial resistance. So we have a lot of microorganisms that exhibit this behavior. So we'll be doing now in the context of bacteria. In order to capture the complexity, the evolving pattern of bacteria, we first of all stratify the model into three submodules. The first one is about human population. Then in human population, we stratify the population into three compartments. We have the first compartment, SH, S subscript H, SH. This one is human that are susceptible to the infection, but they are not infected yet. And then we have another compartment, IS. These are the population that are infected with sensitive bacteria, that is bacteria that can be cleared with the use of antibiotics. And then we have another part of the population that is IR. These are the population of humans that are infected with resistant bacteria. These are the bacteria that can survive in the presence of antibiotic use. So another module for animal population. So we believe that what is happening, the dynamism in the human population happening in the animal population. So what we did first is that we stratify animal population into three compartments. We have SA compartment. These are the susceptible animals that are vulnerable to the attack, but they are not infected yet. Then we have AR. These are the animal population that are infected with resistant bacteria. And then we have AS, animal population that are infected with sensitive bacteria. And then we have environment. We have ES, that is the part of the environment contaminated with sensitive bacteria. And we have ER, part of the environment contaminated with resistant bacteria. And you see a lot of arrows, a lot of lines. These ones are a representation for demographic process, transmission process, recovering process, bacterial development process, bacterial shedding process, as well as persistency in the environment. So most of them are interaction line, some of them are transmission, some of them are recovering, while others are just transition between the compartments. What am I fixing? Okay, this table is description of state variable. When I talk about state variable, I'm talking about those mathematical symbols we use to represent each of the population. So we have SH at time T representing susceptible human, people that are not infected with that infection yet. And we have IS susceptible at time T. So human infected with sensitive bacteria. I believe all of this has been explained already. I believe. So we have force of acquisition or force of infection rather. So when we say force of infection, we're talking about the rate at which susceptible individuals are getting infected. Yes. So the first one is lambda SHS, that is force of infection in humans infected with sensitive bacteria, which we define as the summation of the interaction between the susceptible human, the susceptible human and the environment, as well as infected environment, as well as infected animal with sensitive bacteria. And then we have the other one as a product of the product of fitness costs and the summation of the environment infected with resistant and the animal infected with resistant bacteria. So the force of infection is just the rate at which the susceptible individuals are getting infected and are being transiting to the infected compartment. And the same thing is happening in animal population. So we want to believe that mathematically, the force of infection is bounded between 0 and less than 1. So this we define our recovery parameter as gamma as a function of the antibiotic usage, which we define as a summation of the summation of gamma naught. So this could be a natural recovery plus the use of antibiotic, and the same thing is happening in animal population. So this is a mathematical representation of what is happening in the model diagram. So when we're talking about the S with respect to the T, we're trying to look at what will happen in the long-term run, like the changes in S compartment as time goes on. And then the same thing is happening in each of the compartments. So this is our initial condition. So this table is for the description of parameters. Parameter description. Yeah. Thank you so much.
All right, thank you very much. We'll take one question, then Prof's comments or remark. Anybody with question? No question. Okay, Prof.
Yeah, thank you very much for your talk. I was just looking at it that you have an animal compartment in your model, and you didn't specify the kind of animal. Number one. Number two, I didn't see the relationship between how the animal and the human are having connection. I didn't see. So maybe you explain. Is there any connection? Is there any contact between animal and
Yeah, the contact between animal and human is when human kills an animal and probably he didn't cook it very well and eat it. So he or she can take in the bacteria from there as an interaction.
Also, you specify the kind of animal. So when you say animal, it's too broad. So you should specify. No. So if you say population, then which population of animal? If I say mosquito, there is a particular mosquito I'm talking about. I could not say all the mosquitoes in this world. Just like you have malaria. At this time, you see, that's why your model should be specified. You are not just saying, do you have any answer to that? When you say animal, there should be specific, please. Are you talking about cow, beef, or something? You should be specific. You should not just say animal. If I say animal, elephant is an animal. Lion is an animal. Tiger is an animal. Buffalo is an animal. Mosquito is an animal. So which animal are you talking about?
With respect to that question, I want to believe this is a research work. It's still in progress, and we hope to do a lot of modification.
Biology very well. Okay? Do your biology very well so that you know. Because from what I saw there, it's like your literature, in terms of even location, what are the body? You didn't tell us the body in terms of, because let me tell you something. If I want to fund your work, you should be able to convince me that, what is the situation in Nigeria is killing so many people, mortality rate is very high, the number of cases, even as at last month, was very high. You didn't tell, I didn't see all of that. Are you getting what I'm saying? How do you want to convince me that this work is something that is of interest, even to the government? Let's assume that the government say, at the end of this meeting, you say you did something on antimicrobial or something. Can you show us what you have done? You could not tell us, okay, so these are the things you should go and find out going forward. I'm just giving you some, find out what are the geographical distribution of this particular thing you have. It's different from their own. So it's different. The other group, is it 3 or 5? Group 5 also did AMR. Well, what you are looking at is you are looking at bacterial fitness. Now you should be able to look at what are the geographical distribution of what you are doing in Nigeria. Because we should first stop our own problem before you move to other parts of the world. In Nigeria, what has been done? What do you want to do that is different? Study that. Go to PubMed and do this. So we are giving you till December 15th to send us your publication. We'll be following up on your platform. Thank you very much. Let's start for them. They have tried. Let's have a photo.
All right. While they are planning to take their photo, group 3, can we have your members in position? So group representative of group 3, yes, I have you. I believe their slides can be up. Four. All right. So group 3, can we put our hands for group 3? Let's celebrate them now. Celebrate them. All right.
All right, so group 3, we are working on modeling the effectiveness of non-pharmaceutical interventions on the spread of Ebola Bundibugyo virus in DR Congo. And helping us to do the presentation are Omolola and Adeni. You're welcome.
Good afternoon, everyone. My name is Agbolade Omolola, and I'm here on behalf of Group 3 to present the topic, modeling the effectiveness of non-pharmaceutical interventions on the spread of Ebola Bundibugyo virus in Democratic Republic of Congo. To begin with, Bundibugyo Ebola virus, informally known as Bundibugyo virus, is a member of the genus Ebola virus within the family Filoviridae. It was first identified in Western Uganda in 2007 with suspected cases of 131 and 44 deaths with substantial genetic divergence from other known Ebola viruses. Unlike Ebola virus, Bundibugyo Ebola virus has historically received limited research attention. Prior confirmed outbreaks occurred in Uganda in 2007 and Democratic Republic of Congo in 2012, with more than 98% genomic identity between the outbreaks. Bundibugyo Ebola virus produces the characteristics Ebola virus disease syndrome, including fever, weakness, vomiting, diarrhea, and hemorrhagic manifestations. However, its historical case fatality ratio has been lower than that of Ebola virus. In vitro studies suggest that the comparatively lower pathogenicity may be associated with lower viral replication and delayed macrophage apoptosis. The motivation for this research is due to the emergence of Bundibugyo Ebola virus and continuing uncertainty regarding its transmission, surveillance, spatial spread, and intervention effectiveness. Because of these uncertainties, there is a need to create mathematical models that can evaluate combinations of non-pharmaceutical interventions under realistic epidemiological conditions. The key non-pharmaceutical interventions include infection prevention and control of Ebola Bundibugyo virus, safe and dignified burials of infected individuals, contact tracing, which involves identifying individuals who are in close contact with infected people and monitoring them for symptoms. We also have isolation and quarantine, the use of personal protective equipment, which include gowns, gloves, face masks, and so on to prevent the spread of the disease. We also have community engagement. The mode of transmission of Ebola virus are mostly fruit bats of the family Pteropodidae, and human-to-human transmission also occurs through contact with infected individuals, body fluids, contaminated materials, and potentially infectious disease individuals. The exact reservoir and ecological conditions responsible for Bujumbura Ebola virus spillover remain unknown. The 2026 outbreaks association with an artisanal gold mining area is consistent with the hypothesis that ecological disturbance can increase human-wildlife contact. The research aim is to investigate the effects of non-pharmaceutical interventions in the control of Bujumbura Ebola virus in Democratic Republic of Congo. The research questions are two. The first one is, what is the comparative effect of quarantine, isolation, safe burial practices, and infection prevention and control? The second one is, what is the combination of non-pharmaceutical interventions? What combination of non-pharmaceutical interventions gave the greatest reduction on the burden of Bujumbura Ebola virus in the Democratic Republic of Congo? The specific objectives are to compare the effectiveness of quarantine, isolation, safe burial practices, and infection prevention and control in reducing Bujumbura Ebola virus transmission and disease burden in the Democratic Republic of Congo. And the second objective is to determine the optimal combination of non-pharmaceutical interventions that produces the great reduction in Bujumbura Ebola virus transmission and disease burden in the Democratic Republic of Congo. On this point, I would like to hand over to my colleague to continue from here.
Good afternoon, everyone. My name is Adeniyi Oluwaseyi, here to represent Group 3. So the study area for this study, we are looking at Democratic Republic of Congo, just as we know that Ebola Bundibugyo is currently endemic in this region and is also epidemic in this region as well. So here is a semantic flow diagram for our study. So in this class, we'll be looking at susceptible, exposed, infected, and recovered class, and looking at the title of the research work, which is talking about the effect of non-pharmaceutical intervention on the spread of Ebola Bundibugyo in DR Congo. So for this study, we'll be looking at 4 major interventions, which we have quarantine, isolation, we have quarantine, isolation, and we also have monitoring cases of people that have contact with dead persons, and we also have IPC, which we call infection prevention controls. So for this study, we are looking at 2 classes of quarantine because it is actually a non-pharmaceutical intervention. And the first class is talking about the cases where people are quarantined, but after a while, because looking at the latent period of disease, which is 2 to 21 days, they are noticed that they don't have the disease, so they go back to the exposed class. And the second class is people after being quarantined, there is an evidence of disease presence in them, so they proceed to the exposed class. So they proceed to the isolation class, which is the H class. And we also have a case where we have this death class, where we look at contact with people that have died, because over time, studies have shown that contact with anybody who died of this disease can still continue to spread the disease on the long run. In addition to this, we have some system of equation that actually captures the semantic flow diagram we actually presented. So for this disease, we actually have 2 forms of infection, because the first form of infection is talking about a contact that is being made with persons moving from exposed that are not being quarantined. And we also have the second form of infection capturing the cases of exposed that are being quarantined. So in this first of infection, we actually introduced the 2 other interventions, the 2 other non-pharmaceutical intervention, which is IPC, and the second one, which was safe burial. So the 5 one is talking about the infection prevention control, and the second one is talking about safe burial. So the need for this intervention is to actually lessen the disease burden so that anybody who is infected, either at the isolated class or a diseased person that is infected, will lessen the spread of this disease on the susceptible class. So in addition to that, we have our safe variables. Here are our model parameters, that is parameter that we are used in the formulation of our models. So currently we are in the process of answering some of the research questions. At first, we conducted a scenario analysis to understand the dynamics of the disease on each of the class of that we have in our state variable. We have the susceptible class. This is currently the dynamics. We have the pre-infectious class, also known as exposed class. We have the dynamics in the quarantine susceptible, just like I said that we have 2 classes. We have quarantine susceptible, and we also have quarantine pre-infectious. This is the dynamics of Ebola Bundibugyo on each of those classes. We also have on an infectious class looking at the case fatality rate of Ebola Budugudu in DR Congo, because reports make us understand that we currently have over 46.7% case fatality rates. So this is the dynamics of the disease on the infectious class and also on the infected, isolated individual. We also look at dynamics of the disease on recovered class and also the death individual, understanding that the fact that death individual also contributes to the spread of the disease. In conclusion, we are able to develop a governmental model that was able to capture the dynamics of Ebola Budugudu virus, incorporating some normal pharmaceutical interventions like the isolation that we have as a class, quarantine, which we have as a class, IPC, which is known as infection prevention control, and safe burial practice. And for this study, currently we are able to perform a numerical simulation to just understand the dynamics of the disease. And in the process of doing this work, we will also be able to answer some of the research questions that we have posed in the beginning of the work. Thank you.
All right. Yeah, you can still wait. All right. Any question? One question then. Okay. One, then Prof's remark. Very sharp. Brief.
Thank you very much, Group 3, for your presentation. My question is, you're using non-pharmaceutical interventions. You said 4, isolation, quarantine, IPC, and safe burial, right? But when you were presenting your schematic diagram, you talked about Q1 and Q2. So where does isolation come in, please? Because I didn't understand that aspect.
Thank you very much. So in this flow diagram, we're able to capture 2, which is quarantine, and the H is talking about the isolated compartment. So we created a compartment for those 2 classes. While in the force of infection, we're able to integrate the 2 other interventions, which is IPC, which we bring in as Phi 1 to lessen the transmission dynamics of Ebola Budugudu, and our Phi 2, which is second intervention, which is talking about safe burial, which we intend inducing on the disease compartment to also lessen the spread of disease. The isolation class is what we captured as H. So the reason why IPC is currently in the force of infection, just like what I said, looking at the force of infection, our Phi 1 is talking about IPC, which we induce on the isolated class, because we know ICP is talking about some protective devices that can actually infection prevention and control, because we are able to see that this can be majorly done in isolated places, that is isolated centers where people that are infected or people that are exposed and being quarantined are being placed to. While the third one, which is Phi 2, is talking about safe burial. So we are trying to induce safe burial on the disease, on death compartment classes, just to lessen the kinds of contact people would have with people that have died of diseases.
Thank you.
So thank you very much. But like, I was just looking at your beta 2, beta 1. If you are saying beta 2 is greater than beta 1, then I feel that you should bring out a relationship where you could say that maybe beta 2 is maybe alpha beta 1 or something like that. Something that, do you understand what I mean? And so that you don't just say beta 2. Let beta 2 have a relationship between, express a relationship with beta 1, right? And then, can you, yeah, so you said, I saw the simulation you did, right? So what is the base, and your time is in days, right? So what's the base day? The base, your base day. Eh? Yeah, even if you are not using data, like you cannot just say that I'm just using any base day because you know what is going on. Eventually, you want to use data. Even if you are doing theory, there should be a base day. Do you understand? So you may not be able to answer that now, but you should have a base day from where, find out when did it start in Congo. That should be your base day. Are you getting me now? So you say maybe, I think it's around the data, the one I said something to you now. Okay, the one I did, the one I showed, maybe I think it should be around May, June, thereabout. So, you know, you know, it can only be declared after more than 5. Do you understand? More than 5 cases. So, so like ongoing like that, it was in May. So you should find out the base day So you'll be talking about, so that when you get the data, because I was seeing everything is just like this, like an exponential stuff. So I'm confused with that. You also have to look at your parameter. Where did you get those parameter values used? You understand? Get them and then look at it, whether, you know, so going forward. But we can talk about the data later, because I think I have some data with me. I'm working on that. Okay. So they have tried. It's a good work, right? Clap for them.
All right, pictures. Let's come forward to take pictures. All right, while group 6, where's group 6? Group 6 members. December 15 is the deadline, general deadline for every group 6. Huh? Group 6. Are we ready? Group 6. Let's welcome you. Then after group 6, we have group 7. Group 6 members, let's come forward, please. Let's make it snappy. If you're in group 6, please, let's put it there. Then while our representatives comes forward. Thank you. Group 6 members, let's have you. You are highly welcome. Group 6 members. So who are those who will be representing? Then group leader, you introduce them, sir. Right. So. Yes, sir.
All right, so group 6 will be presenting on modeling optimized strategies for vector control on malaria transmission dynamics across it's still working. Thank you, sir. So modeling optimized strategies for vector control on malaria transmission dynamics across contrasting epidemiological settings, a comparative study of Kwara and Kano states, Nigeria. So the presenters are Mr. Isaac and Mrs. Stella.
Okay, thank you. Like has been introduced, our topic focuses on the modeling, optimized strategies for vector control on malaria transmission dynamics across contrasting epidemiological settings, a comparative study of Kwara and Kano states, Nigeria. Sorry, it's not moving. It's not moving, please. Okay. Now, malaria, as has been established, is a life-threatening infectious disease transmitted through the bites of blood-seeking female Anopheles mosquito. we have that the adult mosquito, in a bit to be able to lay their to lay their eggs, they need blood to suck blood from humans to be able to produce eggs. And in the process of biting humans, to be able to get blood to produce their eggs, if they have contact with an infectious human, malaria, its parasite itself can be they can get the infection from a human that is infectious, and with that, they can go on transmitting it to, other persons as they bite to also get blood. Now we have that, we have that adult vector control with insecticide-treated nets and indoor residual spraying has reduced malaria prevalence globally by 40% since 2000, yet progress has stalled in high burden settings. As we all know, making use of mosquito nets that is popularly known and indoor residual spraying has always helped to, kill some of these adult mosquitoes around us. But that has brought about a very good result in areas that doesn't have very high malaria burden. But in some other areas, the area that is a low epidemiological setting for us in Nigeria, one of them is Kwara. So malaria prevalence itself has reduced there, but some other places like Kebbi that has a very high rate of that malaria infection breaking out there, we have that despite the use of treated mosquito nets, despite the use of indoor residual spraying, we have that malaria burden has remained high in such settings. So it is, however, not clear how complementary interventions targeting immature stages of the mosquito population impacts the malaria burden, disease burden across contrasting epidemiological settings. Now, we are saying that, yes, the interventions that have been coming have been focusing much on the adult mosquito itself, but we have not been able to establish properly how we can incorporate targets on the immature mosquito itself, whether we need to focus on that or combine it with the efforts that are ongoing on eradicating the adult mosquitoes in these different epidemiological setting, and that now relative to resources, because what it costs to be able to provide these interventions for the adult mosquito, we need to keep checking to see if we focus on the immature mosquitoes, are we going to spend limited resources, or it is going to help to bring a best resource. So that is the focus of this study. So this underscores the need for the investigation of the comparative impact of vector control interventions on both adult and immature stages of the malaria vector. Now, while mathematical models often treat vector control as a single aggregate effect, few studies have mechanistically compared adult stage and larval stage interventions across contrasting epidemiological settings in Nigeria, where seasonality and vector species vary sharply. This study therefore aims to determine the optimal vector control strategies on malaria transmission dynamics across two epidemiological settings. The research question is, what is the optimal allocation of malaria vector control resources between adult stage interventions and aquatic stage interventions, alone or in combination in Kwara and Kano State? Therefore, the research objective is to determine the optimal allocation of malaria vector control resources between adult stage interventions and aquatic stage interventions, alone or in combination, both in Kwara and in Kano State. My colleague will just pull it up from here.
All right. Okay, good day, everyone. My name is Olushola Isaac, and I'm here to continue from where my colleague stopped. So I'll be talking about the methodology and the results and some of the things we hope to achieve from this research. So the fact that you already have the aim and the research question at heart. So for the study settings, we're going to be looking at the Kwara and Kano states representing the low and the high transmission epidemiological settings in Nigeria. So those are the two states we're considering. The data we're going to be using will come from the following study: the ITN coverage for each state, routine monthly malaria cases for each state, the demographic data for each state, and the data within existing literature. Then we now move on to the model formulation. So we're going to be using a mechanistic model, which has 10 compartments, 4 for the human population and 6 for the mosquito population. Remember, my colleague mentioned that we're going to be considering the immature mosquitoes, and that's why you have an increased number of compartments in the mosquito population. So you have the susceptible human, the exposed, the infectious, the asymptomatic, and the infectious. You have the mosquito, you have the egg, the larva, the pupa, the susceptible adult, the exposed, and the infectious. So this is the flow diagram that we actually constructed for this particular research. So you can see at the top, you have the susceptible human, the exposed, and the infected, that's infectious. Then you have the asymptomatic, those ones who are actually infectious but do not show symptoms. And that represents the transmission. You can see from the susceptible to the exposed, you have our cause of infection there. Then we can see from the pre-infectious stage, which is the exposed, you have 2 classes they move into, depending on whether they show symptoms or not. So we also have from the infectious, our assumption there was that they can actually get recovered upon treatment. And that's the omega you're seeing there, taking them back to the susceptible class. Then the mosquito population, we have the egg, which is the one that you have the GV there. You have the larva, the pupa, the adult susceptible, the adult exposed, and the adult infectious mosquitoes. So that's the diagram representing the flow that we're going to be using for our research. This is the model equation for the human population. I'm going to skip that. This is also the one for the mosquito population too. Then this also are some of the representations we use from the flow diagram, as you can see there. And remember, we're using the logistic growth for the mosquito, like the recruitment rate for the mosquito, so that we can actually quantify what we call the carrying capacity of the mosquito. And these are the initial conditions we have. These are the descriptions, which I'm going to skip. You can check with us later. Then these are the expected outcomes. So in the course of this research, what do we hope to accomplish? So first is to identify optimal malaria vector control combination per setting. Remember, we're considering Kano State and Kwara State, right? So, and we're looking at vectors intervention, control interventions for the mosquitoes. So we're looking at these 2 settings to consider which intervention would actually be more useful in actually tackling the control of these vectors in each of these states. Then we also have policy recommendations for targeted deployment. So after everything, we have to just let them know that we know what we're talking about, and we give them things that are needed for it. So, and that's it. Thank you very much.
Appreciate them. All right. Yeah, let's do that. All right. So one question. Okay. Then Prof. Okay.
Thank you, sir. Briefly, you talk about modeling and optimization. Sincerely, it's because I like you guys to ask the question. Optimization should be used instead, because if you look at your research gap and your aim, you see that you optimize. So any method you use to optimize will be on modeling. So you don't need to use the word that. And I also noticed that you are talking about Kwara and Kano. It's good. You are looking at the top and the bottom, but you know, geographically, you must look at the south and the north. So in that case, I would advise that you should look at the south, either it's middle or not lower, then you'll be able to have a good geographical location. Thank you.
Very much. All right, Prof.
Okay, thank you. I think what they are thinking is about, there is stratification in terms of the burden of malaria. Now, I think Kwara low burden, and then Kano is high burden setting. So I think it's still fine. The way the burden is being stratified. So we have low burden and high burden zone. So they are trying to compare what goes on in those two locations. So now, you have, so you didn't talk about the data you want to use. I didn't see anything about data. Do you have data for the mosquito too? Where are you going to get the data? Where? Show me on the slide. Where is the data? Existing literatures. Okay, then no problem. You are also mixing LLIN and ITN. So you should stick to, because in the current time, it's ITN that is being, so don't use LLIN, use ITN. We were using LLIN before, but now it's ITN. But another thing is also that now you mentioned that you are using ITN and IRS. Now, what informed the use? Because you cannot just use them anyhow. You know, like when you can't use them at the same time, there is something that must inform the use of IRS and ITN, because the two, they are indoor. IRS is indoor residual spray. ITN is insecticide treated net. Where do you use it? Inside. So now, how do you want to combat the mosquitoes outside? Maybe even indoor, out of bed. Are you getting me now? And then outside. Now, you're talking about different aquatic stages of mosquito. How do you want to tackle them? You didn't talk about the vector that will tackle the mosquitoes, the aquatic stage. You didn't talk about destruction of stagnant water, you know, all those things should be in your work because you are dealing with aquatic stuff. You should talk about other larvicidin and all of that. So you didn't show up in your work. It's not in your model. It's not your model. It's not in your model. So these are things you should improve on going forward. And then this work you are doing, like you need other data more than the routine data. You need other data to make it a robust work. Because if you use only routine data, you have left the core of the work, because the core of the work has to do with vector control. So you need to get data on interventions, on what's it called, coverage of ITN, coverage of IRS, and why should you use IRS when you are using ITN? So you can't use the two at the same time, right? No. If you want to, that's another thing. If you are talking about resistance, it's another thing entirely. According to the epidemiology of malaria, you cannot use ITN at the same time using IRS. You can't. So it's either you are using ITN, and then there is also time bound between them when you are using them, because the two are both inside. Are you getting me now? So these are things you have to still look at on this work. So in other words, we can still talk on the platform as we go forward. Thank you very much. It's a good work.
Pictures with Prof. Then group 7. Group 7, you can come forward. And your representatives. Then after groups, we have group 2. Group 7. Let's have you in front here. Group 7. So I said, thank you. Group 2 is finally ready. Powerfully ready. All right, group 7, let's have group 7 then. Our representatives. Oh. All right. All right. All right. So group 7 will be presenting, and we have 3 representatives. That's the person of Ms. Uwem, Mr. Francis, and Ms. Blessing. Okay, take it over. Okay.
Good evening, everyone. My name is Uwemedimo Ezekiel, and with me are my colleagues, and together we are representing Group 7. Today, we are going to talk of something that is very familiar with all of us here, Africa as a whole, which is malaria. But we are not asking the question, how many people are infected. Today, we'll be looking at a very interesting topic, predicting malaria transmission dynamics under changing climate patterns in different ecological zones. Nigeria is not a one ecological environment. We have the Sahel in the north, the savanna, and the wetlands. And this next slide. We have the Sahel in the north, the savanna, and the wetlands. And this means the relationship between climate and malaria in one geographical zone is different from the other. Malaria remains one of the most important vector-borne infectious disease, particularly in tropical and subtropical regions. It is caused by Plasmodium parasites and transmitted mainly through infected female Anopheles mosquitoes. Although malaria is preventable and curable, it continues to cause substantial illnesses and death because transmission is shaped by biological environment, climate factors, socioeconomic, and system. Nigeria provides an important settling for examining this relationship because of its large malaria burden. Africa as a whole already had the large proportion in malaria, and Nigeria has a very fair share in it. In Nigeria, for instance, the southern coastal regions experience perennial transmissions due to favorable year-round temperatures and high rainfall. Within Nigeria, the prevalence of malaria varies substantially across ecological zones, with the highest coastal regions of the south and lowest in the dry savanna regions of the north. This study is on a systematic review of approaches to developing early warning systems for malaria, revealed that most studies utilize statistical models, primarily using time series analysis such as autoregression integrated moving average and its variants, including seasonal and Sarimax, which accounts for seasonal adjustment and exogenous variables. This study is guided by the eco-biosocial framework for understanding malaria transmission dynamics, which recognizes that malaria transmission is determined by the interactions among ecological factors, biological process, and social determinants. The conceptual framework links climatic drivers, ecological zone characteristics, and malaria transmission outcomes. At this point, I'll hand over to my colleague.
Okay, forging ahead to the research gap, existing studies have established associations between malaria incidence and climatic factors such as rainfall, temperature, humidity, and vegetation, but have largely relied on contemporaneous associations, conventional regression, or location-specific time series and machine learning approaches. Consequently, the magnitude, timing, nonlinear form, and distributed lag effects of climate on malaria and how these relationships vary across Nigeria's savanna and forest ecological zones remain insufficiently characterized. Addressing these gaps is essential for developing robust, ecology-aware and climate-informed malaria forecasting. Moving down to the research aim and questions, the aim of this study is to characterize and investigate the spatial and temporal dynamics of malaria transmission across Nigeria's contrasting ecological zones, with emphasis on differences in malaria incidence, temporal trends, seasonality, peak transmission timing, and inter-annual variability. The study seeks to answer the following research questions. Okay, moving down to the research objectives. The research objectives are to characterize and compare malaria transmission patterns across Nigeria's ecological zones in terms of incidence, temporal trends, seasonality, peak timing, transmission season duration, and inter-annual variability, and to assess and analyze changes in these patterns over time. The second research objective is to quantify the magnitude, non-linear form, timing, and duration of the associations between rainfall, temperature, humidity, vegetation, and malaria incidence across Nigeria's contrasting ecological zones. And the third one is to assess the predictive value and lead time of large climatic and ecological signals for identifying periods of increased malaria transmission risks across Nigeria's contrasting ecological zones. The study areas in Nigeria's ecological zones is given below. We focused on the 8 ecological zones, that is the Sahel Savannah, the Sudan Savannah, North Guinea Savannah, the Dry Savannah, freshwater swamp, lowland rainforest, mangrove, coastal, and then mountain. Moving down to the data description and methodology. 2 kinds of data were gotten to analyze this study. The first one is the malaria surveillance data, which was sourced from the National Data Repository, that is the NMDR of the Nigeria National Malaria Elimination Program. And then the second data was gotten from CHEPS, that is monthly rainfall were obtained from a high-resolution satellite or reanalysis products such as wide temperature and related atmospheric variables will be obtained from ERA5 land. Moving down to the methodology, a retrospective longitudinal analysis was conducted to characterize malaria transmission patterns, generalized additive models, and distributed lag nonlinear models was estimated to determine the nonlinear delayed climate malaria relationships. Machine learning models, including XGBoost and random forest, was used to evaluate the ability to capture nonlinear relationships among malaria, climatic, and ecological variables. At this point, my colleague will continue from here.
Good evening, everybody. I'll be talking about the results of our work. So if you have been following from the start, we are going to answer 3 questions that was proposed. And then from our questions, we were able to bring in 3 objectives. We're able to bring in 3 objectives. And then we're able to, because of time frame, we're able to lay our hands on the first objective and the third objectives. So we are working on ecological zones in Nigeria, and we know that we have 8 ecological zones. Now, from the characterization of the transmission pattern, we did time series and we decomposed the time series using STL. So through all the ecological zones in Nigeria, we were able to see that the Sudan Savannah performs better and then has a strong seasonality. And then the least seasonality is from the coastal region of Nigeria. Also, we try to check the duration, the timing, and the peak. From this table, we were still able to see that the Sahel Savannah has the highest. And then continue from this is a time series of Yobe. Yobe is the state that is represented in the Sahel Sudan that we are talking about. Now, this is the seasonality of the Yobe from the from the time series. Now, also, we were able to lay our hands on the third objectives, and then from the third objectives, we compared two models, the time series model, SARIMAX, and then the machine learning model, the XGBoost. So from our work, we're able to see that SARIMAX performs better than the XGBoost when we check the percentage error. Now, the work is still ongoing. Okay, the performance metrics. So from what the work is still ongoing, and then by the end of the month, we should be able to the rest of the objectives and come up with a better observation. And then by October 2026, we'll come up with the manuscript by grace of God. Yes, sir.
Question? Question? Okay, one question then. We'll take, wait. We'll take one question then.
Thank you, sir. Briefly. Yes. Yeah, I know. You talk about Arima and Sarima in your objective and the like. Thank you, sir. So, and I noticed your work when you are talking about the literature. You mentioned 2023, 2009. This is 2026 and 2025, sir. Work on that. On your, you have to narrow down your research gap. See, from the presentation, you see 1, 2, 3. Otipoju, how to, when to, no. Narrow it down. The more you narrow it down, the quicker you'll be able to achieve the write-up. Then you talk about XGBoost, and you talk about Arima and Sarima. Just Sarimans. Yes, the Sarimans talk about seasonal, like you taught us. But when you now did the model, you told us that Sarima is more efficient than XGBoost. You understand? SARIMAX. Thank you. I want you to do your analysis. I can help you. That XGBoost is going to, I believe, is going to have more efficient than Sarima. Please, I believe so.
Yes, the work is still ongoing, and by the time we finish the whole process, we should be able to check the one that performs better.
So now you have to be careful with starting statements, because this is a research. It's like an ocean. So you cannot really say this will perform better. You have to be careful. That's why we said that when it comes to data work, there are a lot of uncertainty. So what you may be thinking that, oh, don't always think that every machine learning tool will always give you the no. There are other statistical learning tools that can also be of use, that will even perform better depending on the data you have. Do you understand? So that's why you must be careful the way you So now they have done certain part of the work, which mainly has to do with looking at the time series aspect, which for me, I'm seeing it as a good start. Now, the reason is because I told you the other time that for you to really understand your data, you have to work on your data well, look at the distribution, look at the pattern of the data, and then it will inform like complex analysis of the whole data in terms of before you now bring in model. So they have gone through the right route going forward, being able to like come up with the composition of the time series data, and then they have also extended it by using exogenous variable like SARIMAX. That's when you introduce exogenous variable to the SARIMAX. You understand? So which is, I mean, it's a good start. So let's plan for them. So they have also promised, listen, because I'm not going to write it down, but I'm giving them December 15. But they have said they will finish by October 31st. December. Okay. Because I wanted to write it down now, so that I will be waiting for the paper by October 31st. Now you are saying December. No problem. So let's clap for them again. Thank you. Let's come and have the photo.
Yeah, photo. Then group 2, let's come forward powerfully. Group 2.
I would like to invite my wonderful, precious, dedicated, very passionate members of Group 2 to walk majestically up here. All right.
All Group 2.
All right, thank you. Okay, so Group 2 worked on this, modeling the effect of unutilized second dose measles containing vaccine on measles transmission dynamics in Nigeria. Okay, so of the Group 2, 3 of them will be presenting. I would like to welcome Mr. Sahabi Musa, Mr. Falola Sheung, and Mrs. Adeduke Owa Sanoye. Am I correct? Okay. Please, the 3 of you will be presenting for us. So we have the first presenter. Thank you.
Okay, good afternoon. I am Sahabi Musa. I will be presenting along with Mr. Shewan on behalf of Group 2. This afternoon, we shall be presenting our research proposal on modeling the effect of unutilized second dose of measles containing vaccine on measles transmission dynamic in Nigeria. Measles is highly contagious and potentially fatal viral infection, which is caused by a virus called morbillivirus. Morbillivirus is in the family of Paramyxoviridae. Paramyxoviridae are those viruses such as chickenpox, Mumps, rubella. Mostly clinically, we can easily identify them by rashes. They cause rashes mostly and throat laryngopharyngitis. So despite effective, since 1988, there has been effective vaccine for measles, but it remained a global public health concern with 95% of unvaccinated individuals infected by age of 15. So complications occur roughly by 30% when 30% of people that got measles will come down with complications such as otitis media, crop, which is laryngitis, diarrhea, and pneumonia. So case fatality is typically around 3 to 6% in developing countries, but it can be up to 30% among people that are immunocompromised and people from disadvantaged population. There is evidence that link measles to immune amnesia. Immune amnesia is that situation where the vaccine that is meant to save somebody for life, now the waning rate will come very quickly. Measles is second dose of measles. Any child that took that vaccine is expected to be protected for life. So studies suggested that a lot of children halfway, they will have lost all the immunity they got from the measles vaccination. And also it's important to note that there is no specific treatment for measles. The only thing we have is immunity.
So whenever we have immunity gap, definitely we are going to have cases of measles. The treatment we have is palliative, maybe treating the symptoms. Globally, from 2000 to 2024, the vaccination abated 59 million deaths. 59 million deaths. Yet, in 2024 alone, there are 11 million cases and 95,000 deaths occur, mostly among children below the age of 5. So in Africa, Nigeria alone account about 25% of all measles cases. So that is why this research is very important. There are cases of from 212,183 cases of measles. It reduced to 704 in 20, increased to 21,270 in 2025. So this shows that the available vaccine does not really work. So from the WINIC, WHO, UNICEF, estimates of immunization coverage shows that there are a dropout in the people that took measles 1. They normally doesn't come back to take the 56% measles 1 and then 35% measles 2. This is sharp. So this shows that there is serious gap that we want. So the one Nigerian transmission model that we study that took place in Ondo State, that one also did not specifically explain how it bring about, did not observe some of the parameters that we intend to observe. So this brings me to the aim of our study, which is to investigate the effect of unutilized second dose measles-containing vaccine uptake in children who complete measles 1 but didn't currently proceed to measles 2, and how that brings about the transmission of measles in Nigeria. And to achieve that, we designed 2 questions. One, what is the effect of measles-containing vaccine 2 on measles transmission dynamic in Nigeria? And then what is the effect of unutilized measles-containing vaccine 2 on measles transmission dynamic? So to achieve that, we set 2 objectives. One, to assess the impact of measles transmission dynamic in Nigeria, and also to investigate the effect of unutilized measles containing vaccine on measles transmission dynamic in Nigeria. So at this juncture, may I call on Mr. Sheun to proceed with the rest of the presentation?
So as my colleague have explained, so these are the introduction and the research aim and the area we want we have at is Nigeria national data. You want to use so this is the data, this is the weekly confirmed cases, measles cases in Nigeria from 2018 to 2025, and we got this data from NCDC dashboard. So these are model formulation, which is our method. So we developed a compartmental model where we have successive pre-infectious, infectious, recovery, and the first dose and what second dose. What is it called? measles. So for like to address our question now, we discovered that from vaccination coverage, a lot of individuals are taking the first dose of vaccine while we have drop down in the taking of second dose. So that's why we try to look at the effect of the unutilized second dose vaccine on measles transmission dynamic in Nigeria. So this is our model equation, and this is our initial conditions. So these are our state variable and what parameters, description. These are parameter value. So we try to look at conduct two scenario analysis. The first scenario analysis we look at is when we incorporate the second dose of measles vaccine with the first dose, then we look at the effect. Then the second one. So we look at the second one when without what is it called? The second dose of measles vaccine. So in our conclusion, we see that what at MCV1 equal to 71.43%. I'm going to call my colleague to come and round up the result and discussion.
Thank you, Mr. Sean. Good evening, everyone. Please permit me to stand on existing protocol. My name is Adedunke Owasonye, and I'll be concluding the presentation for Group 2. Please, can I have the results? Yeah, we did simulate scenarios with two cases in each scenario. So in the first, where we have a case of those that took MCV1 with MCV2, and the second case MCV2. Now, from our simulated population, in this scenario, we have % of our population that were in MCV1. And in the first case, none of them received MCV2, whereas in the second case, about half of them received MCV2. Now, if we look at, oh no, this is scrolling. Well, we are the last group, so please. Yeah, so if we look at the thick line is representing those that did receive MCV2 after MCV. The diagrammatic representation, there is a sharp consistent decline in the incidence rates of did not receive, where we have an undulating, decline, rise and decline in the incidence of measles, and comparing the two, the incidence rates for cases, there is a significance in the two. We have about 88% difference in the two in the two incidence cases. So for the second scenario, population received MCV1 base, and in the first case, we have those that were with drop, they receive MCV2. In this scenario, all 70% of the case without MCV2 dropout received MCV2. Now, if you look at the graphical representation, it appears like they look similar. Looking at it keenly, we notice a decline, over 26% difference in the rate comparing the two cases. Now, we have discussion for the first scenario. Okay, so anyway, so we noticed that MCV2 vaccination at baseline produced 88.45% reduction in overall measles incidence compared with the scenario without those for MCV for those that did not receive MCB2 for the first scenario, and for the second scenario also, the dropout in MCB2 coverage was responsible for 26.72% of the cases established. Now, key findings: MCB2 utilization at baseline indicates that MCB2 is highly effective in protecting the population vulnerable to infection. We can actually establish that from our result. And also, dropout in MCB2 coverage can imply that MCB2 is capable of providing further protection on the population if the individuals who got MCB1 all got MCB2. Hence, we can say that effort put in place to achieve behavioral change towards uptake of MCB2 can lead to better protection of the vulnerable population. These are our references. Thank you for listening.
Okay. All right. Question, one question, then Prof. One question. Okay, let's take one question, then Prof. All right.
Thank you. I want to appreciate your group. You did a very good presentation. I was that there was this woman from it. That is nice. Your data, you took it from NCDC. That's good of you. Your research graph was precise. No story. That's nice. And I noticed that this work, it was probably this one that put the input, because every of your data, yes. No, it's not assumption. It's based on their presentation. When they are making their word of choices, they put the statistics and they make references to the timing. And the last but not the least, because you did a very good work, 2025. So, but the last thing, however, when it comes to your timing, it's poor. Thank you.
Let's have. You see, please, at the end of this program, we are going to give you a letter of external examiner. So you'll be coming to examine for FOIA free of charge. Thank you very much. So now, one of my comments is, you see, you are on other people's mistakes. Yes, it's obvious. Some of the comments I've made, they are pardons, and that was why they came last. So, yes, it's a wise thing to do. So anyway, like one of the things I observed was like they were stating the statistics, which was very, very good. You understand? If we have a policy maker here, we'll be able to identify like, oh, see, this is how huge or how less measles has affected us in Nigeria. And we could see by numbers. Now, all of us should learn from this. Anything public health, you should present statistics. Give us percent. Let's see the number. It's very, very important. Epidemiology is a discipline of numbers. You don't hide it. Tell us the number. Very, very important. Now, we also saw that, what's it called? They have been able to build their model because, you know, comparably with other vector-borne disease may be quite difficult to track, you know. So because for measles, find out that it's a bit easier, which they have relied on and is okay. Now, one thing I will also try to note is now they have also gotten some results, which is very good. So, which means that the paper can be ready next week, Abi. So, anyway, now, but you have not shown me your model validation before you went to do scenario analysis. So, I didn't see your fitting. I didn't know the method you use in doing your fitting. So, all of this you should put together. But I appreciate all the effort, sincerely. It's not easy. And for every group, I must tell you that from group 1 to group 7, you have all done very well. Very, very well. Because let me tell you, it's not easy to pull people together within a few days and begin to write this, write what's it called? Presentation. It's not easy. You understand? So I appreciate the energy, the passion that every one of us have put into it. Now, one thing I will now say is that this passion must not go down. Are you getting me now? What do I say? It must not go down. So we must drive this passion until we get the paper, until we get the paper what? Published. So if it is not published, it's of 2021 today, it has been in the archive. It has not, as far as research is concerned, it has not been done. Until you publish it, that is when it is done. Yeah. So I'll call them. So until the paper is sent, published, very happy to see. Do you know, I told you the work for last we are written and revise the paper. They have asked for revision. Mogbo, Lassa fever paper, the one we did in Boko, we had about 6 groups. At least 2 groups have submitted, and remaining, I'm still on all of them. They are going to submit before the end of this year. So there is no reason why we should start this kind of work and not finish it. So let's encourage ourselves and finish this work with the energy we have used to start in this place. Let's kick that energy on, and God is going to help us. Let's come now, the group, to take this photo.
All right. So please, let's ensure we stay till the end. We are still going to have a general photo session. And before we close the meeting itself. So, okay, let's group 2 have the picture with our dear director. Okay, you guys smile now. Why are you frowning? Why are you frowning? Is it because, okay. Don't let me talk. Please. 022 22 022. All right, let's have profs. But let's put our hands together for Prof once again.
So I want to, can we get seated? Some of us are outside. Can you come inside? Those people outside, can we come inside? Let's conclude this meeting. Thank you very much. It's been a wonderful time, impactful time. The past 14 days, 2 weeks, we have been here working together, listening to lectures, bringing ideas together, and it's been a time to learn, to relearn, and to unlearn. And I believe that the other, you will have picked several things, not one thing, in the course of this meeting. And apart from the knowledge, you must have also got involved in one network or the other. Now, by collaboration, especially coming from the group level. So one of the reasons why we have groups is also a form of collaboration, networking, where we can continue to do our work, engage ourselves from now. And then let me tell you, this thing we are doing can lead to bigger work, bigger projects, depending on our passion and our interests. Now, we must begin to think in a way of finding solution to the problem in our society. And because when you look at Nigeria, or let me say Africa generally, you will find out that there are many problems to solve. There are many problems to solve. And the only way is that we come together as a united force to solve our problem by thinking through in this kind of platform to be able to find several problems, going from ecological problem, epidemiological problem, problems in the financial space, economy, and banking, and all of that. Now, so some of the things we have learned in the course of these 2 weeks, there are tools in our hands that if we don't go and sleep over them, but we carry on with them and go back and study the people in a particular country called the Barbarians. They went back and they studied so that they did not just assume what they have learned, but they sat down and studied so that they can begin to use those things to solve problems in their environment. Now, we must take an example from such group that have been able to find solution in their own generation. Now, I'll bring us to a point when I studied about a man. Now, the man, in his own time, there was a problem, and the problem was the problem of food security, food insecurity. And the problem was not yet on ground, but it was like there was a prediction, there was a forecast that in few years to come, there is going to be food insecurity where there will be famine. And it's not going to affect their own country alone. It's going to affect several countries of the world. Just like when COVID came, there was an epidemic and many of us were not prepared. Now, one good thing about modeling is it can help us to see the past, or not just see, understand the past, and also forecast and predict the future so that we can make proper plan against any eventualities. So when I read the story of the man was brought in by the president of that country. Now there is to understand how to manage this problem. So this man came and explained the problem. He gave them a light problem. Now, and one thing I would love to know, I would like you to know tonight, as we round up, is that all of us are created to solve problems. Now, you must not be a man, a woman that have the mindset of being a liability. You must be someone who wants to look to your environment and say, what kind of solution can I bring into this? You are in a university, you are in a system where things are not working. You begin to think, how do I solve their problem? Recently, I went to the VC's office and some people engaged me. Sir, what can you do for us? They say, yeah, so we used to gather data for NUC on certain areas in the university in terms of the demography, the student results, the staff, everything about in terms of their educational qualification and whatever. And they put it into data, and it's been very difficult for them to collate this kind of data. So they were asking that, how can we help them? How can the Canada help them? I had a meeting with them, was it this, I think last week. And I began to think of, okay, what can I do to solve this problem and make things easier for them? Now, we must be men and women who are driven with a mindset to bring solution. God has created us and given us the potential to be solution providers. You must not be a complainer. Anytime there is a problem, the mindset must be, how do we go about solving it? Now, if you don't build that mindset from now, you cannot be our next answer to the problem in our environment. So this man I was talking about came to the house of the house of the president, and then on getting there, he was questioned that this problem we are envisaging, how do we go about solving this problem? So the man came and said, okay, I have a solution. The solution is that he developed a model and told them that, look, there is going to be certain number of years of famine from his own prediction. And then there is going to be certain number of years of abundance. Now, within the year of abundance, he told them that in your year of abundance, let there be that when you are getting 100% yield, keep 80%, I'm sorry, take 80% and hit and keep 20%. That's 1/5. 1/5 times 100 is what? It's 20%. So keep 80% for the future. So, and they began to keep that 20%. They were keeping 20%. Now, if a farmer gets like 100 tubers of yam at a time, in their time, they were keeping 20 out of that 100. They were keeping it somewhere. They were keeping it somewhere until, people began to come during the year. During the year of abundance, people were coming. They were getting food. The problem was solved till when the year of famine came. That nation was prepared. The year of famine came. People were coming all over the world. And because that nation was prepared, they were getting food. To the point that now, when the money got finished, people were selling their land to the government. Take our land and give us food. Take our tax and give us food. And at the end of the day, we saw that the model that man developed worked. Now, what are we saying? Now, I want us to go out there as we are living this with a mindset to go and solve problems. And how do we go about it? Some of the things I've learned in this place, don't sit down and say, oh, I've learned it and leave it like that. Engage it. Now, I sent a student to Senegal some years back. I wrote him a recommendation and he went to Senegal and he was sponsored by SIMPA. So he got there. It was a 2-week program. He got to the program. They gave them a good accommodation, feeding, everything. He was enjoying. In fact, he came back and told me that, oh, Oga, we chop. We chop. Anytime he's talking, he always talk about the food. I said, this is good though. He said they even went to the Embassy of Nigeria. That the man that was there gave them money. I said, wow, that's good. So at the end of the whole meeting, I told him, okay, some of the materials you got from that meeting, can you send them to me? Send them to me. And then I still have them in my laptop to date. That was roughly like maybe 7 or 8 years ago. Yes. So I tell those man, I started reading them. I saw some areas like stochastic model applied to epidemics. I read it. That was the first place. The second time I'll be reading that. I read it cover to cover. I consulted textbooks. Everything that he brought from that workshop, I read everything, all the slides. And then I started researching stochastic. I published paper, first paper. In fact, one of my papers won an award. I got, was it $500 or $600 some years back. So, but the guy that went to CHOP in Senegal did not publish one paper. Do you understand me now? So it means that when you go for workshop, don't just go there and chop. Do you understand? Some of you, you leave the work and hold on the shop because it's workshop. Abina, so you go and begin to shop, go and do shopping. Some of you are doing shopping, buying. I see many nylons and everything, doing the amala. Very good. But let me tell you, as you are going back, you need to engage these materials. Sit down with them. Think of how you are going to use those tools to start doing something. When I was coming up as a young academic, that was if I go for any conference, let me tell you, everything I'm teaching you today, I also learned them. But do you know that most of those places, those places that were doing this program before, the funding is not there again. They are not doing them again. Those programs, in those days, I used to go to a place in Ghana, in Italy, ICTP, they will sponsor. But today, the funding has gone down. In fact, one of the things they were saying to us that in those days was that, Emmanuel, as we are teaching you, take this thing back. Some 10, 12, 14, 15. The first time I learned R was 2012 in Tanzania. 2012 till now is how many years? 14 years. When I got back to Nigeria, I told one of my friends, I said, have you heard about R? He said, no. He went to their office in UI, their department. He said, sir, do you hear about any program called HAR? The lecturer said, man, get out of my office. There is nothing called HAR. Yes, but maybe 4 or 5 years after, the man came back and told them, there's a program called HAR. You see information. Now, let me tell you, there are certain information you have gotten now. Information. Some people say information will make you what? It will make you fly. Yes. You will not walk. That's why now when you are flying from here to Abuja and you are going by road from here to Abuja, is there no difference? Is there no difference? When you're on the road, how many hours will you spend from here to Abuja? Maybe like 12 hours. But if I fly from Ado to Abuja, the next 45 minutes I'm in Abuja. So that's the difference between a man that has information and a man that does not have information. So I want to challenge you. You have been loaded with many information. But do you know that it's not enough? Now, 3 weeks or 2 weeks training, we are still going to have virtual meeting. 3 weeks training, 2 weeks training is not enough to make you a machine learning expert. 2, 3 weeks training is not enough to make you a mathematical modeler. So don't let me see you on your LinkedIn. Machine learning experts, data science experts, international exposure. This, that. You go and write it. Yes. Many of you, you see, there was somebody we taught like that in Abuja, some medical doctor. We just taught them. And the next thing we saw, under 10 months, mathematical modeler. I was like, just 10 days, how many weeks? 10 days of contact. You don't become modeler. Okay, now. So please grow. And one of the reasons why we put the platform together is a means for mentoring. Now, after this, you need mentorship. You need to grow. And how do you get mentorship? You need contacts. That means further contacts. Don't just say, I met Professor Bakari, I know him. We took pictures together. It's not enough. Do you understand me now? So we need to now say, okay, maybe there are times especially from the group, the paper you are working on, create this thing there. Let's do this work. Let's finish this work. You'll be learning. As you are doing it, you are learning. You are getting understanding. You are looking at it before you know it. When I first learned R, I went somewhere. It was just 2 weeks, but I didn't stop there. After that time, I started using R. I taught people R. In fact, those days, I did workshop in Boye here on R. I trained people. I started training my students. I exposed people to her, a lot of people. Why? Because I start down to learn it further. So you also have that capacity if you are ready to sit down and learn it. If you are stuck, ask questions. Sometimes you will fall down. You will rise up again. That's the way to do it. But if you say, oh boy, well, this thing is too hard, then you will not be able to go further. All of these things we are learning. Let me tell you, one thing that can make you an expert is constant repetition. When you keep repeating, you keep reading, you keep checking, you keep working, you keep doing this. Do you understand? You will become an expert. And then don't run from mentorship. It may not be towards a PhD. You may need like, can you come, sir, can I come and spend 1 month in the do some work? We can arrange. Oh, can I do this with you? Can we do this with your center? Can we do that? And then from there, from time to time, and then when you see us having webinar, like now, this evening now, I have put up the flyer of September, our first webinar answer into that platform. You see there now. And then you can register and be part of that kind of thing. And anytime there is training for anything, get involved. You keep learning, and from there, you become an expert. Now, you see, some of the guys that were used, but not some, all of them that were used to give classes here, they are all my students. They are all my students. And none of them, when they were coming, came with what they were teaching. It was while we were coming together, mentorship, under mentorship. I have to say, oh, this one is going now. Come and teach this. There are some of them, this is their first time of coming to this podium. This is their first time. Because we have seen them grow. We can present them now. When we also see you grow, we'll bring you from Nasarawa, from Bono, from this. Come and be part of the facilitator. That's our expectation. So we are planning that in all the 6 geopolitical zones, we should have representative. Everywhere should be a cube in Nigeria, not one-sided. That's our plan. So that if there is a epidemic in the Northwest, I will call. There was somebody from Northwest here. Northwest. Kano. Northwest, Northeast. KB, Bono, Kano. There were some people, maybe they have left. North Central. Nasarawa. I will say, what's your name? I don't forget your name. What are you doing? Send data to us. Come and work. Let's look at the Ebola that is coming there. And we send it to WHO or NCDC. We come together. All the North Center, we sit down. There's a problem. We have done this kind of, bring that method we have used during our workshop. We do it. Before they know it, they will bring your picture on NTA, CNN. Modelers from North Central. That's all. That's the way you start solving your own problem. That's what we want to see. So please, let's go back with that burden and passion to go and also work on what we have learned. And I pray that God will grant us wisdom, knowledge, and understanding. And God will also give us long life so that we enjoy all our labor. I want to use this opportunity to appreciate all of you that have waited till this time and that you believe in us. That's why you have stayed true. We appreciate you so much. And we also believe in you. And we also hope that we will work together for long. So we also appreciate all our facilitators. Can we clap for them? You can see the way they are passionate about, they are giving out, dishing out the stuff. Now, the next workshop again, we'll still bring other new. This time around, you will see more ladies, more females, because we want to be gender balanced. Some of you females that are hiding, we are going to bring you up to this podium. And you come and teach classes. You will stand there and teach art. because we want everybody to know how to do it. So some of you that are hiding now, we are going to bring you home. You are going to take classes. Some of you will be assistant facilitators. You are going to do something, and from there you also grow. We want large number of people to also be part of this. Some of you that has come for the first time, next time we see that, oh, we are now doing something, we'll bring you. Come and teach for us. We'll pay your transport. Okay? So let's do it. Thank you very much. I wish you journey mercies for those of you that are coming back tomorrow. And for those of you that are going to stay back, I also wish you a wonderful stay. One thing you must not forget. Now, we have many things in Nigeria, many good things. But you know one thing we lack? The fear of God. People don't have the fear of God. And I want to challenge you as you go back. The fear of God is the beginning of wisdom. Now, where there is lack of fear of God, there will be evil, or evil will multiply. We must do everything we do with the fear of God. Are you a lecturer? Have the fear of God. Are you a medical doctor? Have the fear of God. Are you a nurse? Have the fear of God. Are you a teacher? Have the fear of God. The fear of God is the beginning of wisdom. Anybody, any nation that their leader does not have the fear of God, that nation will collapse. There will be more corruption. There will be more sickness. There will be more disease. There will be more problem. There will be more. Today I was reading that Kenya says, the Kenya president said that I'm going to give free education to all public universities. That they will not be paying school fees again. I want to see when that will start in Nigeria. That any boy that go to public university will no longer pay school fees. I want to see when that will start in Nigeria. I think Nigeria should lead in that kind of thing. Some of us who are young, I want to challenge you. Do you also have the fear of God? Some of you are challenging the president of the nation. In your own small space, do you have the fear of God? Are you not cheating the exam hall during the exam? Are you not stealing government property? Check your heart. Do you have the fear of God? When they keep money in your hand, how do you do it? How do you keep the money? Some of you are a treasurer. You cannot keep 100,000 naira. You have started stealing from it. And you are condemning President Oba or Tinubu. It's not going to work. Because in your own little space, you are not doing your own part. So please, I want to challenge us. If we want this nation to be great, it's not just going to be by education. It's not enough. You need the fear of God in your heart. Your heart must be equipped with the fear of God. Education is not enough. We have had people with education. They have not helped us. They have PhD, they have MMD, they have HIV, they have NYSC, they have HPV, everything. Yet, the problem is not solved. The problem is not solved. We keep on recycling the thing. The problem is that we don't have the fear of God. If you have the fear of God in your heart, you will know that even when you are doing something in secret, there's somebody that sees you. Even when you are thinking it in your heart, you will know that somebody is seeing you the way you are thinking about it. And that thing is captured. You will not want to do it. We need the fear of God to paddle this nation forward. Let's carry it in our heart as we go on. We have gotten a grant. We got a grant here. And by the grace of God, we have used it the way God wants us to use it. I was telling somebody, some people came here from another country. And they were asking us that we should give them I should write to them that they paid $500 for registration. About 4 of them. Because the reason why they were doing that was because they know that Nigeria is a corrupt country. You cannot go to a white man and tell him that one. So they wanted to test it. So they came to me. I was standing out there in February. They said, please, can you? Yeah. So what you just do is that I will tell them I want to collect $250 out of it. So $250 from 4 people. How much is that? $1,000. I'll put that in my pocket. I signed to them. You register 500, then they will chop the 250. I look at them and say, be content with what you have. Me, I have conducted this program free of charge. I cannot write anything like that for you. They were shocked because they know that Nigerians are corrupt people. So by the time they were going back to their country, they went back with another mindset that it is not all Nigerians that are corrupt. Now, they were the one that sent Lasmi. You see that lady, that Indian lady, they were the one that sent him and said, call Professor Bakare, he will bring you, because of what we did. Let me tell you, anything you are doing is going on record. Are you getting me now? Anything we are doing is going on record. Now, what we did, the man was telling everybody, everywhere. That was why a lady from India will not come to this place, because they have bad information about Nigeria. But because of the trust, the man, the professor, told him that call Professor Bakare, the place is good, he will take care of you. And then, so that's why any recommendation you want to get, you must start with truth. Truth. Righteousness exalt a nation. Sin is a reproach. Thank you.
All right, can we do that better? Thank you so much. And that's, for us, it's a very beautiful one, because you know that the heart of our director, by God's grace, is one that has been yearning, that this truth should travel across all the nations, and effort has been put in place to ensure that happens. And today we are all witnesses to all that is going on. And so I would just want us to take our photographs now. There are still information for us, please, particularly against next week. But let's take our pictures first. Hello. Media. Where's media people? Yeah. All right. Now, please let's listen. Lastly, for those of us who are staying back, you are staying back for the next week from now till anytime next week. Please quickly see Prof outside. Prof is waiting for you. For those who are not traveling, who are going to be here physically, you are not traveling for the next between now and next week. Please quickly see Prof. Prof is waiting for you, please. If you are traveling back, no problem. You don't need to see Prof. But yeah, the hostel is still available, and this venue is still also available. Yes, this venue is still very much available for us. The hostel is still very much available.